Hi, welcome to the last full week of our course. So, like I said in the introductory video, now that we have learned about genetics, chapters 12 and 13 are all about seeing how this knowledge can be and is being applied into many different applications, especially on healthcare field. Another thing that I want to mention is that the material that we are going to cover in this and next module is the same as in your textbook, but we will be placing a little different emphasis on it. You can think of it in this way. Look at the number of intended learning outcomes on these two modules. So yes, these chapters are important, but there is a lot less than for the previous chapters. So these chapters are more about seeing why you have to learn about all that stuff. So let's start with defining what is biotechnology. Well, it is the use of biological methods for some technological or industrial uses, and we will be looking at a few of these in just a moment. And the genomics studies the entire set of all of the genes that make you the person you are. So it is all of them, not just some. Now that we know about genes, we are much better equipped to realize also that we can use and potentially also manipulate them. And this is what we call genetic engineering. When we take some genes that we cloned into a greater numbers and add them into an organism. And this can be either the same organism or a completely different organism. We will see some examples of that in this module. And just to be sure about the terminology, just as a reminder, remember that cloning is when we make identical copies. In this case of a certain gene or genes that we are interested in. This is so that we have a lot of material to work with. And it really is amazing what we can do with these approaches. And we will be talking more about DNA recombinant technology using an example of producing human insulin in the next item of this module. But for now, let's talk about the polymerase chain reaction. This is often abbreviated as PCR, and you will see that abbreviation being used quite a bit. And what happens in PCR is that we take the original segment of a DNA that can carry, for example, the genes that we are interested in, and then we amplify it in great numbers. This basically means just making a lot of copies of it. Well, how do we do it? Well, we had DNA polymerase enzyme, which we mentioned in our last chapter. And for the purposes of how PCR machines work, it is important that it can handle high temperatures, as these machines use heat to separate the two DNA strands. And primers are what we use to select the specific sequence that we want to copy. It is the starting point of where the target is. We do not want to waste our resources in amplifying everything possible. And then we make a copy on both strands, so after each cycle we have doubled the amount of DNA. And this makes really quickly a huge number of copies. I mean, after the first cycle we have moved from 1 to 2 copies. And the second cycle from 2 to 4. And third cycle would result in 8, and the fourth in 16, and fifth in 32, and by the end of sixth cycle we already have 64 copies of this sequence. So this is a lot and very fast. So how do these PCR machines work then? Well, there are many different kinds, but this is one. and I have this in my lab. So they are not really unfeasibly expensive, and what they do is exactly what you saw just a moment ago. The sample is put in with some enzymes and other needed chemical components, and we heat and cool the sample, with each cycle making more and more of the DNA sequence of our interest. So it really is not technically an insanely difficult process. There is, though, certain knowledge that you will need to have to be able to do it successfully. And the reason why you do want to know a little bit about PCR is that it is something that is used in most of these applications of biology or biotechnology that we will be discussing about. And there is this great video that I have also included in this module's activities. It's a bit of a 90s pop song for geeks, but you will want to know it for the short answer questions. And one of the applications that you might have seen of this is DNA fingerprinting. And this is what they always do at the TV shows such as CSI and what other ones there are. I do not watch a lot of TV, so I would not know, but occasionally I switch channels and see how they are so-called running a DNA fingerprinting, and I always cringe when I see it, and I will tell you exactly why in a second. But what DNA fingerprinting really does is that it compares individuals. And just like you have unique fingerprints, your DNA will be unique. And there are two techniques that we can use: gel electrophoresis and short tandem repeat profiling. But basically what we do is that we need just a tiny amount of DNA from the crime scene, and then we use PCR, this method that you just learned about, to increase the amount of it. And we collect samples from all of the suspects and amplify the DNA on those samples too. And if the segments that we study match for the suspect and for the crime scene, we can conclude that both samples are from the same origin. And now we just have to find out why and or how the suspect's DNA ended up to the scene. But do notice that DNA fingerprinting, like much of the work that we do in the forensic field, does not prove someone as guilty. It just places an individual to the scene. But what bugs me a little bit about these TV shows is that I actually have worked in the forensic science field, and in these shows it always looks like they put in a sample into some bizarre machine, press a button, and out comes a print that says guilty. Well, it does not work quite that way. Actually, PCR takes some time, and the machine would be only able to tell us if it's a match. But I guess that it would be kind of boring to watch a TV show where for hours the DNA analysis is just stuck on the in-process stage. Anyways, I just find it somewhat silly. Another thing that we see on this figure is this guy holding a gel where he has run these DNA fragments, and there is a ladder that he uses to study their length and so on. But really these days, much of this analysis and reading the results part is done on an automated process on a computer. But if you were going to go into this field, it would be important for you to understand the very basics of how it works. So, what does DNA fingerprinting allow us to do? It allows us to do many things. We can detect many things, for example, that an individual is more likely to develop a condition of some sort, or screen an unborn baby in case there are some major issues that may affect parents' choices whether to carrying through a pregnancy is feasible, or if you are predisposed for certain cancers, for example. I think that Angelina Jolie was found to have a higher risk of breast cancer due to her genetics and therefore went through a double mastectomy. We could also be scanning every single nucleotide of an organism, but this would take a long time if there is a lot of genes. For example, for humans, it took 4 years to scan the complete sequence of nucleotide base pairs, and this work was done simultaneously in parts by many research facilities around the world. So really it is not usually done unless there is a good reason or an organism has only a short DNA or so on. What else can we do? We can also identify individuals, as we saw with the DNA fingerprinting, but it does not always have to be a crime. For example, who the parents are can be easily identified in this way and is actually done quite often. And then, of course, the species that are evolutionary close to each other have more similar DNA. Remember those evolutionary trees that we looked at the beginning of this course? So, closer to organisms are on that tree, the more similar their DNA would be. So often when you think about an evolutionary biologist, you might not be thinking that they work with this high-tech DNA stuff, but absolutely they might be. So next I have a few interesting activities for you to have a look of, and then we will move on to talk about cloning. And I have a good story to share there.